Welcome to the 8th episode of Lab Rat Chat. This podcast is dedicated to informing the public on the importance of animal research and how it's 100% necessary for continuing to make medical advancements for both human and veterinary medicine. Lab Rat Chat is supported by Americans for Medical Progress through the Michael D. Hare Fellowship, which is a fellowship awarded annually to support projects that inform and educate the public about the critical role of animal research in furthering medical progress. The fellowship honors late Dr. Michael Hare, a renowned board-certified laboratory animal veterinarian who dedicated his career to scientific and medical advancements, and who was deeply committed to animal welfare and advocacy. So for anyone that didn't listen to our last episode, well, first of all, go listen to it. There's some really great information about the coronavirus and the serological and antibody testing that we hear about every day in the media. And second of all, just know that Danielle, again, isn't able to join us for these episodes, but she will be back as soon as we can all resume our normal lives. On today's episode, our guest is Dr. Ricardo Carrion, a professor and researcher from the Texas Biomedical Research Institute in San Antonio, Texas. And his lab works with some of the world's most deadliest viruses using very strict safety protocols, and he breaks some of this down for us on the show. And as so many labs are doing, he's working with the SARS-CoV-2 virus and is hoping to find information and data that will hopefully lead to groundbreaking discoveries and help us all beat this pandemic. So sit back, relax, and enjoy this 8th episode of Lab Rat Chat. Welcome, everyone, to the 8th episode of Lab Rat Chat. Today, we have a fascinating guest from the Texas Biomedical Research Institute that you're all going to enjoy hearing from. I know I will, but there's something I always forget to do at the beginning of our episodes. And for those of you out there who may not listen to all of our episodes to the very end, like you should, you may not know that we're giving away 5 $100 Amazon gift cards. And to enter to win one of them, all you got to do is leave a review on Apple Podcasts, iTunes, Stitcher, or Podchasers. And we say those ones because I believe those are the only ones that allow you to leave reviews for podcasts. And then also for some of these gift cards, we're going to look into doing some alternative ways to gain entry points in order to win the gift cards. So just stay tuned for that. We'll announce those details through Twitter. So make sure you follow us at the Lab Rat Chat to stay in the loop on everything we're doing. And then also email us at any time at our email at labratchat@gmail.com. And so on to today's episode. As I said, our guest is a professor and researcher from Texas Biomedical Research Institute who works on vaccine development, amongst, I'm sure, many other things, with some of the world's most deadliest viruses, including the SARS-CoV-2 virus, which causes COVID-19, we're all more than well aware of at this point. So thank you for joining us today, Dr. Carrion. And if you could, just tell our listeners a little bit about yourself and your background and kind of how you got to where you are today to your position at Texas Biomed.
Great. Thank you again for the invitation to meet with you today. We're excited here to talk about what we do at Texas Biomed. Our business is eradicating disease, so we're very proud of the work we've been doing over the past almost 60 years, actually more than 60 years. So I'm a researcher at Texas Biomed. Texas Biomed is located in San Antonio, Texas. And I started research in graduate school actually looking at parasites. So currently my specialty are viruses. So parasites are much bigger than viruses. And I was looking for a parasite that caused a cutaneous sore in South Texas called leishmania. I then transitioned to looking at a virus that infects leishmania during my PhD work called leishmania virus. And then during that time, in 2001, we're all aware of the events of 9/11 as well as the anthrax attacks. I became involved in studying anthrax, the plague, and then eventually Ebola virus. And that's when I became hooked with viruses in general. So since time, my focus has been primarily developing models to try and test efficacy or the effectiveness of vaccines and therapies against these agents for which no cure. So I mentioned Ebola virus that kills up to 90% of people infected. Marburg virus is a similar way, Lassa as well. So I studied all of these viruses with the intent of making models, usually non-human primate models, to study the disease itself. So currently, my focus is, and you mentioned this, working with a SARS coronavirus 2, which causes COVID-19 in humans. We are looking for monkey models that can perhaps replicate the disease we see in humans. Again, the goal of that study is to try and develop models that we can use to test vaccines and therapies prior to going into humans and human trials. So if we show effectiveness in these monkey models, we will be able to use that model to make predictions on what type of vaccine or therapy would be effective, what doses would be effective in humans. Now, we have experience doing this because since 2004, we've been working with Ebola virus. And I mentioned to you previously, Ebola virus is a highly fatal disease for which there are no cure or vaccines up until now. And we at Texas Biomed are actually part of the team that helped test some of these vaccines and therapeutics during the last outbreak in non-human primate models. So there is a candidate drug that came out that we were part of doing some of the preclinical studies. So these are studies in animals that showed which combination of these therapies at doses would work best in non-human primates, which was then translated to human trials. So using that same experience of model development, working with non-human primates, we applied that experience to now the COVID-19 story.
That's pretty fascinating stuff. And I totally understand getting kind of hooked on that. When I first got into research as well, I worked with vaccine development stuff with anthrax. We shared a lab that was doing research with plague. And we also collaborated with doing some aerosol studies with Ebola as well. And I'm in veterinary school now, as our listeners know, and I'm trying to, after school, get back to be a veterinarian within high containment laboratories and get back to that kind of work because I love it as well. And so I planned on kind of touching on this later on, but I think since we've had the introduction to these deadly viruses and even some of these bacteria such as anthrax and plague, how do you guys work with a virus such as, the SARS coronavirus 2 or Ebola or anthrax or plague, which are bacteria, but then some of those deadly viruses where there is no cure? How do you guys work with those without infecting yourself, your staff or the environment or, bringing it home or just out into the communities? So is there pretty strict standards and procedures out there for that, huh? And even the laboratories themselves are pretty secure. Can you just talk a little bit about that?
That's correct. So to study these viruses, pathogens, they're usually classified with regard to the type of disease that they cause, the ease of transmissibility, as well as whether or not there is a cure or a vaccine. So essentially, viruses for which there are no cure, for which there are no vaccine and can be easily transmitted in the lab, causing high mortality, for which Ebola virus is essentially the prototypical BSL-4 viruses. Those are called biosafety level 4 agents. And that's the most safe laboratory for which one can work with agents. For that, in addition to special controls within the laboratory regarding negative airflow into the laboratory, redundant systems, individuals are actually suited into a spacesuit type uniform. So it's a single continuous jumpsuit, if you will, that has a zipper, and a worker will climb into that suit, zip it up when they're tethered to a breathing air supply line, and they work in a BSL-4 laboratory in that suit. In addition to the actual physical presence of the suit, we also adhere to very strict laboratory procedures, such as working within these biosafety cabinets. So these are cabinets that help contain any type of aerosol. So if you're spinning a sample, for instance, if you're spinning a blood sample down, that mere action of centrifuging a sample could generate an aerosol inadvertently. So we have procedures in place to prevent that by using aerosol-resistant or aerosol-tight canisters, doing some of these manipulations within a biosafety cabinet, which is another form of a containment. So that way, if there is an aerosol, it's not spread into the rest of the room. So that's what we rely on, are both the PPE, personal protective equipment, like the suit I mentioned, the laboratory facilities themselves using negative pressure in the case of this BSL-4, along with tethered breathing air, and the procedures in the lab. So that's level 4. Level 3 viruses and bacteria are those that cause severe disease, but mortality may not be that high. And there is a cure, vaccine, or some sort of treatment that can help you get through this infection. So this would be anthrax and the plague, for instance. Those are level 3 level 3 pathogens, primarily because there are therapeutics, antibiotics work against those. There are vaccines for some as well. And now SARS also falls into that same category. Now, the main difference between BSL-3, PPE, and BSL-4 is that instead of getting into a single-piece suit that's zipped up and you have an external breathing supply line, for us at Texas Biomedical and others, it means that we have a disposable, semi-permeable Tyvek suit. Similar to what you see the hazmat units use as white material. Also, painters use it to protect themselves as well. So it prevents any material from getting onto your skin. But the key factor in this is that individuals wear a respirator. So we use a powered air purified respirator, which filters out the air prior to going to your face. And it creates an area of positive pressure around your mouth and nose so that when you're working in the laboratory, you're able to breathe clean air without getting any type of contaminated air. Also, these N95 masks is another way that some individuals adhere to this strict precaution of protecting their respiratory route. Again, this filters out any type of bacteria or viruses from the air so that you're able to breathe clearly. Also, in these environments, you wear protective eyewear, double gloves, booties, and then everything gets disposed of on the way out. So that's a level 3 laboratory. And biosafety level 2 laboratory is something akin to what you would see in a college setting or somebody working with E. coli. So these are pathogens for which they can cause disease, but the disease is mild. It's not necessarily that easily transmitted. And usually without any type of intervention, a person will get better. It's like, for instance, whether just regular E. coli, if you were to drink that material, you get diarrhea, and then after a couple of days, resolve it. So something along those lines where you can resolve it, but it is a concern because it does cause disease. It's just not at the same level as a level 3 or level 4 type disease. So those how we classify pathogens. And a level 1 organism is kind of like the paramecium you see in high school lab classes where you look under a microscope, you see a parasite that doesn't cause any human disease. So that's like a level 1.
Gotcha. Awesome. Thanks for breaking that down for us. So basically, the BSL-4 lab is what you see in the movies, like Contagion and Outbreak and all of that, where you're in the lab with the big spacesuit on with the air, as you just so described. Definitely sounds like you guys are well protected in there, and even double down on some of that and just say that, all the air is redundantly filtered. Anything that goes down drains goes to decontamination systems that are heated to super high temperatures or incinerated even in some cases, and before they go out back into regular sewage pipelines.
Yeah, thanks for pointing that out. That is correct. So nothing actually comes out of the BSL-4 laboratory that hasn't been autoclaved. Not same for 3, autoclaved. Also, what I left out of the BS4 laboratory, there is a chemical shower. So as a person wearing that spacesuit exits the laboratory, they are required to stand in a chamber, which is filled with atomized disinfectant for about 8 minutes, and that decontaminates the suit. So when they step out of that chamber, essentially the suit is clean. So that's an extra step that's unique to BSL-4. Where the suits aren't disposed, but they're cleaned with a chemical disinfectant for a prolonged period of time.
And you can't even get out if you wanted to without going through that shower, in most cases, correct?
It's correct. They're interlocked. They require the shower to run before you can exit. So that is a good point.
So before we get into some of the research you all are doing with this coronavirus, you've mentioned a little bit, but could you just tell our listeners about what Texas Biomedical Research Institute is or who you all are? We talked with one of our episodes, 2 episodes ago now, with the Tulane National Primate Research Center. And I saw on your website, it mentioned the Southwest National Primate Research Center. So are you guys associated with them? And we talked about some of the national primate research centers in general and how you guys work in collaboration and as far as data goes and helping to solve problems and cure diseases by sharing that information with each other. And that also helps with the refinement and reduction of animals and all that. So maybe if you could just touch on the relationship there with the Primate Research Center, or if you guys are one and the same, and just about Texas Biomed in general and your guys' research goals and what you aim to do.
Yeah, so Texas Biomedical Research Institute was established back in the 1940s, and the goal was to do this type of innovative research. Our purpose would be to develop now therapeutics, vaccines, and our goal is to eradicate disease. So anything associated with that is within our mission itself. Now, the Primate Center, the way that comes into association with Texas Biomed is the institute, the facility. The Southwest National Primate Research Center is a grant awarded to the institute to host this Primate Center resource. So the Primate Center is on the campus of the institute, but it is essentially the institute. It's just a division of the institute. So the biosafety folks, everything else supports the Primate Center as well as those that aren't associated with the Primate Center. The main issue, as you pointed out, or the main objective is to be able to provide or have these non-human primate species as models for the nation to use. So us, along with the other Primate Center, our goal is to be able to develop new models, have available models. These are even complex diseases that can utilize these models. They'll be available for research.
So getting into the animal models and the research you guys are dealing with as far as COVID-19 goes, I read in a press release you all put out, I guess it was 3 or 4 weeks ago now, about using different monkey models, but also using mice and guinea pigs. So I take it you have other species there as well besides just monkeys. In addition, you guys are also doing work with baboons, which I guess falls under the monkey model. So can you just tell us about the research initiative you all are doing and how studying the virus maybe in multiple species at once will help in our understanding of this coronavirus?
Yes, as you're aware, this SARS coronavirus 2, the virus that causes COVID-19, came out of nowhere. So it was something that we needed to approach aggressively. And at the time, it was unknown if monkeys were going to be good models or what type of monkey is going to be a good model for the disease. So our approach was our president actually sought out approval from the board. So we're a private nonprofit institute. So essentially, we are reliant on money through grants and contracts to be able to sustain our research. We're lucky in the city of San Antonio, we have a good base of citizens that donate to the Institute. And those monies allow us to have buildings and to support pilot studies. Well, our president asked that we set up an association where we're able to get donors to provide monies to actually help support some of these pilot studies. Again, as you know, being in this field, any type of work with a non-human primate increases the cost significantly. So it's not possible to do this without support. So through that, also we have a partner through a commercial company, as well as a BARDA, which is a part of the Health and Human Services. We were able to get monies to look at animal models. And the three models we chose were the baboons, the rhesus macaques. Those are old world species of macaques, and then a new world species called a common marmoset. These three different species of non-human primates we used to infect with SARS coronavirus 2. And the intent was to try and be able to induce a disease state that was similar to humans. The reason why we want to do that is because if we were going to test a vaccine or therapeutic, we wanted to do it in a model that was more relevant to humans, so had a similar immune system, but also caused a disease that we can show if we were to give it a drug, we can have some sort of benefit. If we were to give it a vaccine, we can prevent infection. So that was the rationale for choosing these three non-human primates. Aside from that, there's also an interest in developing some small models, rodent models. And these are, again, to help facilitate some of that early testing. If you have a small animal model, you're able to screen many more of these compounds once you identify in vitro which ones would be good candidates to go further prior to putting them in non-human primate models. So overall, you reduce the number of non-human primates you need. So that's a second arm of our approach. So we have the non-human primate model development being one aspect of it. And then these smaller models would help to support any type of drugs or therapeutics that would come through our facility before going into a non-human primate model.
Yeah, very well thought out. I think that's one point that we've driven home and one thing we've tried to make clear is that these animal experimentation protocols that come through and the ICOOK reviews and all of that, they're well thought out. There's a reasoning for everything. Yeah, the numbers are always refined and reduced wherever possible. So that's great to hear. One thing I want to touch on, and we kind of talked about in our last episode as well, is it's called the SARS coronavirus 2. Is there a reasoning why the virus is named that? Was it a mutation of the original SARS virus that we saw in 2003, if that was even the original? And then just the kind of, maybe some of the logic behind the name. And then if you guys did work with SARS, and do you have data from that that's applicable to developing a vaccine for the coronavirus that we see today? Or did they develop a vaccine back then? I know it wasn't as deadly and widespread, obviously, but at the time, I remember it still kind of being a scary thought, but then it went away rather quickly compared to this one.
With regard to the nomenclature, I haven't looked at that too intently, but I believe, based on what I understand, it's very similar to the original SARS 2003 outbreak. So there hasn't been, as far as I know, a vaccine for that. There was another one, coronavirus MERS, that emerged in 2012, Middle Eastern Respiratory Syndrome. It was a coronavirus similar to SARS and COVID-19. So the technology and the approaches used to study these viruses are benefiting us now because we are able to use some of the same type of platforms to address SARS coronavirus 2. Additionally, I think the advantage we have from the standpoint of model development is the investment in animal models that looked at Ebola virus, for instance. What needs to occur within the laboratory to make data obtained from these animals very consistent with what's obtained through a clinical trial. The quality systems that are required, how we characterize viruses that go into these animals, how do we characterize assays we use to measure parameters associated with the animals. We learned all this through helping to get the Ebola vaccine and these therapies online. And we're applying that same approach to this SARS coronavirus 2 situation. I think that's going to help expedite any type of vaccine or therapeutic because we do have that acquired experience working with other viruses. So to your question, yes, I think both the SARS experience with SARS and MERS has helped us to be more prepared to deal with this in a rapid way. So one thing that really stands out about this SARS coronavirus 2 epidemic, I don't know if you touched on it with your previous podcast, was that the level of collaboration seen in this outbreak, this pandemic, is greater than any other type of collaboration I've seen in the 20 years that I've been doing science. There are working groups nationally, also working groups that the WHO has set up on. I serve as part of those working groups where data are freely discussed in these forums. And overall, it leads us to a more rapid understanding of the disease process, as well as more rapid development of vaccines and therapies. And I think now the help of technology has enabled us to communicate more freely. And also, we are all affected by this common issue. So there is an urgency and a need to collaborate more than ever before.
Yeah, the level of collaboration from everything I've read too has been truly amazing. I'm not currently in school right now, not in the lab, but just everything I've read. So whether in some labs just switching their roles to help focus on how they can help in studying coronavirus as well. Another thing I wanted to bring up, or since it's been a few weeks now since I've read this press release, how far into the research are you guys with developing an animal model? Have you found any exciting or interesting data with this coronavirus? I've read a little bit about infecting the primates and after they recover from the disease, reinfecting them, and then they're not getting infected again. I mean, that's encouraging news. Have you guys seen anything like that? Or you been looking at that? Or what kind of exciting data have you guys had so far, if you're able to, of course, share that at this point?
So right now we are actually doing analysis of samples that were collected during the study. But we're seeing what's been published by others is really a lot of the virus shedding occurs early on after infection. So we're characterizing that more closely, looking at samples that were collected during the study. So we're very early on. But our goal, again, was to look early on after infection, what happens, with the intent of trying to use this model to see if therapeutics or vaccines could prevent or cure the virus. So I think what we've done, we'll be able to put forward very soon a paper that describes these findings.
Okay, great. Well, before we wrap this up too, I wanted to touch a little bit on some of the misinformation that's being published out there regarding animal research and developing, a useful and effective vaccine or therapeutics for coronavirus. There's been multiple reports out there, especially early on, that, researchers for this coronavirus aren't needing to use animals. They can go from the lab, from in vitro trials, from in vitro experiments straight into human trials without evaluating the product in the animal model, which just kind of blows my mind that those articles are being published out there. I think everything you've said here today neglects a lot of that. And can you just shed some light? I mean, these animal research models are still 100% necessary in order for us to develop vaccines and therapeutics, right? There's no workaround at this point for coronavirus to allow researchers to go from bench work, in vitro cell culture stuff, or computer models into a human trial. Is that correct, or am I misspoken?
No, that's absolutely correct. I mean, in order to understand how a vaccine or therapeutic works, you need to have a relevant living system that mimics humans. So the complexity of these viruses, they take over your machinery. So essentially, they're a coded piece, in this case, RNA, that enters a cell and takes over it. So when you're looking at developing therapeutics and vaccines, it's very important that you get that right, because if you're injecting this into a human, you can make the disease state worse. There's a way that you can induce an immune response that might enhance disease. So in order to determine if you have a drug that you can place in a person, you have to evaluate its safety, first of all, in a relative animal system, whether it be a monkey or something else, but efficacy for sure in a model that replicates disease and is similar to humans. Otherwise, you can have these unintended consequences where we think we develop a vaccine, we show a immune response with antibodies, but it turns out they're the wrong type of antibodies. And instead of curing the disease, you created a situation where you enhance disease. So instead of seeing the 3% mortality rate, now you're seeing 30% mortality rate. So again, we're not at the point now that computer models or other type of in vitro systems can predict that. You need to have a system, in the case of vaccines, can reproduce the immune response. And even some of these other type of antibody-based drugs, again, you need to test those in a monkey or non-human primate model to see if even giving that antibody-based drug doesn't have any sort of antibody enhancement occurring and resulting in a more progressive type of disease. So long story short, we still need models. Again, our goal is to reduce the number of animals that are being used. We have several statisticians working on each type of experiment to make sure we use the fewest number of animals. We refine the techniques to cause as little pain as possible to these animals during the course of a study. And we also maximize samples taken from these animals. So for instance, if I were to take a serum sample now, we can use it for genomic analysis, looking at CBCs, chemistries, as well as using it to test diagnostics and therapeutics, looking at cytokines. So we maximize every piece of information from these animals so that we can, in the future, be able to reduce the number of animals being needed for future studies.
Awesome. Yeah, thanks for clarifying that. The reason I wanted to bring that up again was just there's a lot of those articles that are talking about fast tracking the vaccine. And then you see a lot of the comments and people talking about how, that means they can skip the animals and how the animals are no longer needed. So I just wanted to, we've hit on that obviously a lot on this show, just to bring that point home once again. Even during all of this, you still need animals. I'm just wondering, if you said you did work with the MERS, did you guys do work with the MERS coronavirus at Texas Biomed as well?
No, we did not. We did it with the SARS coronavirus, the initial one. We were looking at these inhibitors of replication. Back in 2014-15, we published the work on that. It was using a mouse model.
Okay. I was just curious because I remember reading back in the day when that was going on about one of the labs working, the troubles and difficulties working in a BSL-3 laboratory using a camel model. And I don't know if that was something you guys were doing there. That just blew my mind when I read it. We talked about all the different animal models out there, and this lab actually had camels within their high containment laboratories, which is pretty fascinating to hear about.
Yeah, that's a lot of work to maintain. Yeah, we do non-human primate models. We don't do any type of livestock or anything like that.
And this kind of last question, we ask all of our guests this question at the end and just ask them to kind of describe what they say to people when they ask what they do for a living, whether you're on an airplane or social gathering, a stranger somewhere that you're talking to. Do you bring up the animal research component of what you do when you're describing what you do to them? What are their reactions to that? Or how do you approach, that situation and handle it professionally to be able to describe to them if they have concerns about the animal research? Because it just kind of helps a lot of listeners out there don't always know how to talk about what they do in a way to help people, understand without setting off the bells and whistles in someone's head about, your animal research. You think of animal experiments and animal testing and all the pictures that you may see online. So how do you address that out there when you're talking to somebody?
So generally, when they ask me what I do, I let them know I'm a scientist and I develop therapeutics and vaccines for agents that cause disease in humans. And then they'll ask what type, and I mention Ebola virus, and I'll let them know that we have a dedicated group of people that help us to test vaccines in animal models, and that these animal models are important for us to advance these therapeutics to a point which they can be used in humans. So essentially, I'm from Texas, and most of my individuals here in Texas, I don't know if we're spoiled we're in San Antonio, Texas, so this is a military city. Everybody understands that there is risk in things that you do. So we have a BSL-4 here, and right across from us is a Chili's restaurant. So there's really no concern about what we do. But they also understand that there is a need to use non-human primates and other models to help advance technology and these therapeutics. Fortunately, there is a wealth of information online about how to do an animal study. There needs to be a tremendous amount of oversight. There's committees that are available. And if the topic comes up to animals, I just specify that every study we do is reviewed by a committee that's mandated that every animal's life is what we consider important. And everybody in my team values animal life. I mean, if they didn't do that, they wouldn't be part of this team. And the way they care for these animals, like their family members, is something that I think creates a better experiment because we have happy animals, healthy animals, and the care for these animals makes what we do even greater because we're doing it with care and we value the life of these animals.
Yeah, that's 100% correct. And I like how you related how you talked about Texas. One of the many things I love about Texas are the people down there. We lived down there and for a few years right out of school down in Galveston. And it was a great experience, great time in my life. So before we finish this up, is there anything else you want to tell our listeners? Any final statements or thoughts that we haven't previously covered that you think the public should know regarding humane and ethical animal research about this coronavirus pandemic in general?
Because of the current pandemic, we all have to do our part to help prevent the spread. So again, that means social distancing, wearing these masks when you're in public places. If you're sick, isolating yourself. I think that as fellow inhabitants of this earth, we're required to do that. So I think that's something that's been spoken of. And not everybody gets how important that is. Because even though we may be healthy, others aren't. And if we get them sick, there's 10% or greater, 18% chance they're going to die from this, where will probably be okay. So again, I think it's looking out for other individuals, which is important. And also don't start drinking any type of household cleaners to try and cure yourself of diseases. You have to use approved FDA drugs or therapeutics and not go off label on these things. That's important as well.
Yeah, great points. One other thing I neglected to mention, how far off are we looking before we see a vaccine come out for this, you think? I mean, we're still looking, you hear about it every day in the news, it sounds like, but it's still probably next year sometime, right?
Yeah, I think that's right, but that is accelerated. So if you look at the vaccine development, and looking at Ebola, for instance, as an example, it was about 7 to 10 years after a really good product that they were able to license that vaccine. So that in 1 year, we're talking possibly having an FDA licensed vaccine for COVID-19. It's pretty remarkable. I think, again, it speaks to the fact that we learn from these other studies, these other agents, and we're able to refine the process for developing vaccines. We have things in the pipeline that could potentially work against other coronaviruses that were able to be adapted. So again, 1 year for a vaccine from a virus that didn't exist before December 2019 is a pretty good track record.
It's pretty incredible. That's for sure. Thank you again for coming on and speaking to us today and providing a wealth of information. I learned a whole lot. I'm sure all of our listeners will as well. I'm going to reiterate the $100 Amazon gift card here. Again, leave a review on Apple Podcasts, iTunes, Stitcher, Podchasers to be eligible to win that one of those gift cards. And we're also going to come up with another way, I think, to give away some of those gift cards. But again, just follow us on social media. Our Twitter is @TheLabRatChat. And if you have questions, concerns, comments, topics you want to hear us talk about on the show, email us at thelabratchat@gmail.com. And thank you again, Dr. Carrion, for coming on the show today and speaking with us. We really appreciate your time during this busy schedule that you have.
You're welcome. Thank you. Appreciate it.